Hi, I'm Professor Paul Hoskerson, and this short video is about running the ActDES database. This is work from Jana's PhD in collaboration with Nelly Selem and Paco Barona Gomez through a long-term collaboration that we've had. And when we were looking at expanded gene families in actinomycetes, we built this very useful database. Whenever we talked about this at meetings, people often asked us whether they could run their protein sequences through the database. So we made the database available on Figshare and also via GitHub along with the code, and then we published the associated paper earlier in 2021 in Microbial Genomics. If you wish to look at any of the background, you can find that here. So the background for the ActDES database, we created the initial database by looking for highly curated, high quality genome sequences of actinobacteria. So there were 614 genomes that deemed to meet our quality of a low number of contigs and high quality genomes. All the details for this are in the ActDES paper, and this spans 614 actinobacterial species across 80 genera. So this is partly because it has a good phylogenetic spread across the phylum, but is overrepresented in genera like Streptomyces, which are overrepresented in the database simply because of the interest in this group of species. So these 614 genomes were reannotated in RAST, and then this allowed us, in the initial work that we did, was to download the subsystems and count the expansion of particular gene families, and that was published in 2018 around pyruvate kinase. But the database as a whole is much more useful to the wider community, so you can download the protein database and then you can search this custom database yourself. You can also search for nucleotides, and you can create your hits and then build an alignment, and then you can either use something like QuickTree to build your tree, or you can do Bayesian analysis. So much of this is very flexible and you can use any programs that you're familiar with, but I'm going to run through a simple way of taking our FASTA protein database, building a custom BLAST database, searching that BLAST database, building an alignment, refining that alignment, and then building a tree of that alignment. So what you'll need to run the analysis is a version of Bioconda or miniconda running either on a virtual machine or a Linux machine or terminal on a Mac, whichever way you run your command line work. And then there are two data files available, the all protein sequences.fasta and the all nucleotide sequences FASTA. And you can download these from Figshare, which the link is there. I realize it won't work in the video, but I'll paste it below the video, and you need a copy of JALView, which you can download from that URL as well. And then all other aspects, we'll go through the installation of each of the different programs needed. I also have a GitHub page which has all of the code detailed as well. You've installed a version of miniconda onto your computer, then you can check what present working directory you're in, okay, and then you can move to the directory where you have your data stored for what you need. So in terms of my computer, I have this stored in a folder called Bioinformatics in my normal user directory, and you can see that the two files that you need to download from the Figshare account are the all protein sequences FASTA file and the all nucleotide sequences FASTA file. Okay, so if you migrate to the current working directory that you need, Okay, so CD. And you can see when you list those files that the all nucleotide sequences and all protein sequences FASTA files are in that directory. So you're working in the right directory. Okay, so there are a few packages that you need to install to allow you to use the ActDES database. So you can use the package manager Conda to do this for you. So first of all, we need to install BLAST. So this will take a few seconds, but you can use that install command and you can proceed as normal. This will take a few seconds to work through. Watch me install a whole range of programs. So you will have installed BLAST. If you can install muscle using the conda install -c Bioconda muscle, install gblocks, which will help us clean up the alignments later on. Need to install a hammer utility. This will allow us to change the format of some of the files using the easel utility. And then we'll also need to install QuickTree in order to build our phylogenetic trees later on. And again, using the standard conda install commands for those. You can start to do the ActDES database building. So every time you download a local installation of BLAST, you need to build that local database. So you can use the command, which is make blastdb, and then you tell it what your input file is, which in our case is going to be protein sequences, so all protein sequences FASTA, which is in your directory above. Then you tell it which database type, which is protein, and then you have to parse the sequence IDs. I've spotted a mistake in my command, which is there. Okay, and then you can click enter, and it will start to build the new database. And this will take a few minutes to do this. So that took almost 90 seconds to run in terms of building that BLAST database, and you can see that the files have all appeared up in that directory that we had before. Okay, so it's added 3 million sequences to that BLAST database from those 615 genomes. Okay, so the next thing to do is to search that database. So I've downloaded a FASTA file of the protein that I'm interested in. So this is SCO2494, which is a PPDK enzyme from Streptomyces silicola. And we're going to search that local database by using the following command. So blast p minus query, okay, and then your input sequence. So in this case would be SCO2494.FASTA. Okay, so that's the sequence I downloaded from SCODB. And then you need to tell it what the database is. So the database is all protein sequences .FASTA, and then we need to specify the cutoff values that we're going to use for our proteins. So the E value, we'll set that at 0.1. This is where you'll need to hone your searching of the database, and you can only really do that by blasting the database and then going back and looking at the sequences that you retrieve from your BLAST database. You're going to tell it that the output format is 6. So if you look in the manual for BLAST, this just tells you that it's a tabulated database. And then we're going to tell it that the maximum number of target sequences to retrieve, and we'll set that at 10 million, which is more than the database contains. And then we'll pipe that into a cut. And the reason for doing the cut function is it just takes the first 2 columns in this part of the database. And then we'll tell that to create SCO 2494, and we'll call that hits. Okay, missed a space out there, and then we'll just call this a .text file. Okay, so we can hit go, and that's reading the database now, and it will flag up a few invalid residues in part of it, but it will just continue to search that database. And again, this will take a few seconds for it to come back with the hits. It's completed now, only takes a couple of minutes, and then we can just check to see what those hits look like using the less command. So we can put in less, and then you put in your file name, which we called SCO 2494, and we call the output file .text. So we call that hits, didn't we? And there you go. You've got all of the hits. So you can scroll down through this, and you can see that you've got all the hits in Streptomyces, you've got them in Frankia, etc., and you've got lots and lots of hits. So you can hit Q, and that'll return us back to the terminal window. Okay, and then the thing we need to do now is use the sort and the unique function to take those files, sort them into a list in order, and then identify any duplicates that have ended up in that database. Okay, so we can call this sort, and then we can give you an output file name, which would be your SCO 2494 underscore hits.txt. Okay, so it's going to sort the file that you generated from your BLAST. Okay, and then what we can do is we can pipe that and use the unique function to identify all of the unique sequences, all the unique lines within that file, and create a new file that we can call SCO 2494 underscore hits, call it underscore unique. Okay, we'll make that a text file. Okay, and that takes a second, and that will parse those sequences, sort them, and then remove any duplicate sequences. And again, you can use the less command to look at the SCO 2494 underscore hits underscore unique.txt, and there you can see that cleaned up file that's within your set of sequences. So you press Q, and that will take you back to we've got this unique list of sequences that are our hits from the BLAST database, the SCO 2494. We can then use the BLAST command sequence again to extract those sequences from the database. So not just the sequence IDs, but the actual protein sequences. Okay, so we use BLAST DB command. Sorry, they're not uppercase. And you tell it the database type. Protein. Okay. Database was from all protein sequences dot fast A. Okay, and then you want to tell it that the entry batch is derived from your SCO 2494 hits underscore unique. Okay, .txt. You want to create a new file. Sorry, txt. Want to create a new file, which will be a FASTA file because it's got your sequences in, and the output, we'll call this SCO 2494 underscore hits underscore unique. Okay, but at this point we'll call it a FASTA file. This should then pipe that out as a FASTA file. It'll take a few seconds to do, and it'll skip a few where you've got duplicates or it can't find them in the database. And this appears to be a continual problem with this Hygroscopicus subgingangensis strain for some reason, and I'm not sure why. And these Venezuelan strains are missing as well, but it doesn't affect the overall database. And then we can look at the less command with SCO 2494 hits unique dot fast A, and what we should see is the FASTA sequences of the proteins in that file, and you can see that there. So each identification number, so the accession number for the strain, the name of the strain, and the protein sequences there. So you can check through that, and you can use Q to go back to the terminal window again. So once you have your list of hits from the BLAST sequence and you've pulled them with their accession number and name and the sequence of the proteins that you're interested in, it's at this stage that you can then use the muscle program in Python to align this. And the easiest way to do this is to use the code lines for muscle, which is muscle, and then you specify your input file, which in our case is the SCO 2494 underscore hits unique dot fasta file, and then you specify your output file for the alignment to go into, and you can call this SCO 2494 alignment, and it's at this point you need to save your file as an aligned FASTA format file, so AFA file. Okay, and then you can run this alignment command, and this should take a few minutes in order to do this, and this will vary depending on the number of hits you have in the database. Whilst this is running, what you can do is you can download a program called JALView, and this will allow you to look and edit your alignment. So that will take a few minutes to run. And we'll come back to that once it's run, and we can go through the output file as it basically parses the sequences and starts to align them. Alignment is now finished, and you can see it took 1 hour and 43 minutes because there are 1458 sequences in that, and you can already see that the minimum length of those sequences is somewhere 57 amino acids in length, which tells you they're improper hits, and this is where you need to go back and refine your sequence inputs and your BLAST parameters to make sure you're getting the sequences you require out of the database. The average length of the sequences in the alignment is 700 amino acids. So the easiest way to edit and look at this alignment to see how it's functioned is to run a program called JALView, which you should have downloaded. So you can open JALView, and you can open your .afa file that we created through the alignment. So you can tell it to open all files, and then you can open your alignment, and you can see in JALView that you've got all of the sequence names, and then you can look at the conservation, the quality, and the consensus sequence for your alignments. And you can already start to look along the alignment and look at the quality of that alignment. So by removing those unusual sequences that we talked about earlier that are very short, you can go through the file and remove those, and the alignment looks pretty good in certain sections, and this requires a lot of manual time actually looking through and manually curating not only the sequences in your file, but also checking that the alignment has gone to plan. So once you've done that, we'll assume that this alignment is fine just to progress to the next stage. But this often requires you to go back and iteratively reprocess and realign sequences once you've edited the sequences out, such as those short ones that we talked about earlier. So we'll assume that that alignment is fine, and we'll move on to the next part of the ActDES database. So we run a small program that is called gblocks. And gblocks is essentially a computer program written that removes poorly aligned positions and divergent regions of alignments, either in DNA or in proteins. And this just tidies up the alignment and removes anything that might then later skew the tree making programs. So again, it's a fairly simple set of code to run through the program, so hopefully you will have installed this earlier on. So we can start to code for gblocks. So gblocks, and then you put in your output file, which is your alignment file. So it was SCO2494 underscore hits. Sorry, we call it alignment dot AFA, which tells you that it's an aligned FASTA file, and then we want to add in a couple of little parameters to help you do that. So B4, and you can look at the documentation for gblocks. I'll put the link below. So B4 is a way of telling it what the minimum length of a block would be, and we can set that at 5. B5 is how many, or how the gap positions are allowed, so you can either have n for none with half of the gap positions available, and because we're doing a large scale evolutionary analysis, it's worth going with half for this, or A for all, and you can modify this depending on how you want to go back and rerun the analysis. So it's B5H, and then B3 as part of the code allows you to look at the number of contiguous non-conserved positions, and you can kind of create any integer for this. And for our purposes, we'll choose 10 again. You can alter these parameters as you see fit. So then you can create a log file for this, which will then save, and you can view this if need be. So you can run that script, and it's very quick, and you'll see that the log hits has arrived up at the top. So finally, is in order to be able to build a tree from our alignment, we need to convert our AFA file into a tree file. And the tree file is a format called Stockholm. And at the start, we download the Hammer program. And within the Hammer program is a little set of utilities called ESLs. Or I guess the bioinformaticians had easel in their mind when they created this. So we can just use the reformat tool within easel, and you run that program by running ESL reformat Stockholm, and then you put in your aligned FASTA file. So Stockholm is telling us what format we want, and our alignment file is called SCO2494 underscore alignment AFA, and we want the output file to be SCO2494, and we can call this tree file, and we'll call that STO for the Stockholm format, and then that should run relatively quickly, which is done, and then we can start to run the quick tree program. So hopefully you will have installed quick tree. Again, we can code this using quick tree, and the input is A for amino acids. The output is the tree, and then you can tell it how much you wish to bootstrap. So you can tell it to bootstrap this 1,000 times, and then we need to tell it that the file that we're going to use is SCO2494 underscore tree file dot Stockholm, and then we want to call the tree SCO2494 underscore actually we can just call that dot tree. Okay, Now this takes a long time to run. Okay, so the quick tree has run and we've gone back to the command line, and you'll note that in the ActDES directory there has been the SCO2494 tree file created. So now we need to view this, and there's several tools that are available to view and visualize trees. The one that's simplest to use is Interactive Tree of Life, or ITOL. So we can create an ITOL file. So go to My Trees and you can click Tree Upload. You can select your file to upload and it will take you to the folder where you stored your files, and then you go down to your tree file and then you can click Open. And that will store that. So ITOL allows you to store your trees in projects, and then you can click Open and it will build the tree. And then so you have many of these options that allow you to alter your tree and customize the tree, and also to export the tree in any format that you like. And you can see that all of the nodes are present, and at the end you can see the mouse over each of the strains. You can also change the tree into unrooted trees, which is one of the better ways to see duplication events and some more of the features that we might be interested in for this. Okay, so with that, this is the end of this little tutorial, and hopefully that was useful. Cheers.